It's one thing to copy our DNA to store our genetic information, but it's another thing entirely to actually do anything with that. While DNA is able to maintain steady sources of genetic information, RNA is the route that we actually use to copy that information into a useful form as proteins. We call the copy it from DNA to RNA transcription. The RNA will eventually carry that information to our ribosomes to be translated into proteins. We already have talked about two different major differences between DNA and RNA. Try to remember what those are by looking through your notes. Here are some other ways that DNA and RNA are a little bit different. RNA is almost always single-stranded, and because RNA is single-stranded, it cannot error check in the way DNA is. It's not a good long-term storage for genetic information. RNA is much smaller than DNA, and RNA tends to have many different functions, and we'll see many of those functions through the transcription and translation processes. There are 5 major functions of RNA that we'll see throughout transcription and translation. The first of these is heterogeneous nuclear RNA, which is formed directly from DNA transcription. HRNA would be a direct copy of a gene on the DNA, the gene being a segment of DNA that codes for a specific protein or group of proteins. We oftentimes talk about the genome, which is the entire set of genetic information. It's the collection of all available genes. So the heterogeneous nuclear RNA, it comes directly from a gene and is copied. But the heterogeneous nuclear RNA is not converted directly into a protein. It actually first gets sliced into smaller pieces so that only the useful information is brought. The smaller pieces are messenger RNA, and the small nuclear RNA are what help to cut the HNRNA to the mRNA. We'll also see during the process of translation, the ribosomal RNA is required to actually form the ribosomes, the enzymes that allow us to create proteins. And the transfer RNAs are going to actually do the translation, the interpretation of the RNA signal into proteins. And we'll talk in more detail about how each of those work. The first step of this process, transcription, is actually really similar to DNA replication, except that now we only need to make one copy for the RNA. We don't need to make 2 copies, and the DNA will rewind rather than separating into 2 strands. This means the hRNA is produced all by itself, and the DNA is maintained as permanent storage of that genetic information. After the hnRNA is produced, the hnRNA is converted into mRNA. The hnRNA consists of 2 types of pieces, both exons and introns. Exons are the types of pieces that will actually eventually give information for a protein, and the introns are unnecessary pieces for that signal. Those pieces, the introns, need to be removed, and the small nuclear RNA are the ribozymes, like enzymes but with ribonucleic acids, that are able to remove those sections. When the exons are joined together, they become a messenger RNA. Now the hRNA are copied from genes, but not everything that is in a gene is turned into a protein, and not every protein from the gene is the same. We can take a set of 4 exons, like is shown here, exon A, B, C, and D, and we can actually splice them together many different ways. We don't always have to include all of the exons. That means that many times we can create more proteins from a single gene than just a single protein, which means that the transcriptome, the complete set of all mRNAs that can be produced from a genome, is actually much larger than the genome itself because of all the possible alternative splicing patterns. Now that we have messenger RNA, we need some way of taking the RNA nucleotide sequence and converting it into an amino acid sequence. To do that, almost all organisms use the same universal genetic code, a matching of 3-letter sequences to specific amino acids. In general, don't memorize these codes. However, I do want you to know what the start codon is, or the initiation codon. In RNA, the initiation codon is AUG, which matches an ATG sequence in DNA. Those codes are interpreted through transfer RNAs, or tRNAs. tRNAs are bound in the ribosomes so that the anticodon section, a 3-letter sequence on the transfer RNA, is able to match on to the codon section of the messenger RNA. Each of those transfer RNAs has a specific site to attach an amino acid that is specific to that transfer RNA. The 3-letter sequence of a transfer RNA anticodon is able to match up with a messenger RNA codon in such a way that transfer RNAs will line up next to each other and bring the amino acids that are attached close to each other in order for them to react in sequence as well. But how do the amino acids get on the transfer RNA in the first place? We accomplish these with specific tRNA synthase complexes, or enzymes, that are able to take a tRNA that matches with a specific binding site for that enzyme and bring it close to a specific type of amino acid that gets charged or loaded onto the transfer RNA. After the RNA is transferred onto the amino acid, or the amino acid transferred onto the RNA, it can bring the amino acid to the ribosome to be synthesized. The ribosomes are actually enzymes that consist of both protein and RNA that together perform the translation of the proteins. So ribosomal RNA is part of the ribosome enzyme. Ribosomes consist of both a large subunit and a small subunit that both work together. Because the ribosomal RNA plays such an important role in this, we call it a ribozyme, an enzyme that is an RNA. This is actually our second example of a ribozyme, the first being our small nuclear RNA, which helps to cut our heterogeneous nuclear RNA into the messenger RNA. Proteins at the ribosomes are translated in 5 steps: activation, initiation, elongation, termination, and then post-translational processing. We'll walk through each of these steps in the next slides. Activation is where the tRNA synthase attaches an amino acid to a tRNA. This actually happens outside the ribosomes, but is required to constantly be happening while enzymes or protein synthesis occurs so that we have a steady supply of charged tRNAs. After activation, the mRNA will be bound to the small subunit of the ribosome at an AUG sequence, remember that's the start codon, in the messenger RNA. A matching tRNA is going to bind and bring along an amino acid, in the case of the start codon, a methionine, and a large subunit along with it. So now we have a single tRNA bound into the ribosomal complex at what we call the P site. The next step is elongation. That's the process by which amino acids are added to the chain one by one, each time forming a new amide or peptide bond. After the first tRNA is bound, a second tRNA binds according to the next 3 base pairs, making sure that its anticodon will match the codon in the messenger RNA site. As that bond happens, a new bond is formed between the first and second amino acids, and the first tRNA is then released. The ribosome moves down the mRNA and binds onto the next tRNA. The process continues over and over, each time elongating the chain of amino acids by 1. At some point, we need our protein to stop being formed. Termination is the process by which elongation stops and the protein is released. It occurs when specific stop codons, UAA, UAG, or UGA, don't memorize those, appear. There are no tRNAs for these stop codons, so they signal protein release. Post-translational modification can now occur after the protein is formed, or even actually while the protein is being formed in some cases. The types of post-translational modifications that we see include things like cutting off the first amino acids. Oftentimes the methionine is not required, or other amino acids are produced that help the protein to fold, but then are later cut out. Disulfides also form at this time, helping to stabilize the tertiary structure of the proteins. At any time we have quaternary structure, we need multiple peptide units to come together, which means it has to happen after we've produced multiple peptide units. Many ribosomes can participate in the translation of one mRNA at once, so the ribosomes can actually bind at multiple locations along the mRNA, which means that we can use a single mRNA to make many, many copies of a protein. Now, we've talked about this before, But we have 5 major functions of RNA, and now we've seen how each of these are used in the processes of transcription and translation. Heterogeneous nuclear RNA is the first copy that's made. Small nuclear RNA cuts that copy into messenger RNA, which is actually the piece that will be translated into protein. The piece is translated in the ribosomes, which consist largely of ribosomal RNA, and they're interpreted by transfer RNAs that bring along the correct amino acids to the translation sites. We've talked a lot about how replication, transcription, and translation work when things are going right. Some things do not go right, and mutations are an example of that with DNA. Mutations are errors that are made during DNA replication that result in the changing of genetic information. Usually having 2 copies that are complementary results in having an ability for our cells to check whether there are errors, and when they relatively rarely happen, they're usually fixed. Sometimes the DNA is actually changed. A common example is when a mutagen is going to change the structure of a gene. Substances can be mutagens, but we can also have UV light and other ionizing radiation be mutagenics. When a mutagen changes the structure of a gene, that then changes the proteins that are produced from this. And usually this is a bad thing. Positive mutations are fairly rare. So we rarely get things like Teenage Mutant Ninja Turtles or superpowers. It's a lot more common that we get something like cancer. Another way that DNA or RNA can end up going wrong is in viruses. Viruses are small particles of protein and DNA or RNA that need a host to replicate. They don't have any of their own central metabolism. This creates huge problems for our immune systems because the viruses are going to inhabit our cells themselves and use our own central metabolism. We treated a lot of pathogens with inhibitors that can target specific aspects of the pathogen central metabolism, but when the central metabolism is our own, we can't kill off the virus without killing off our own cells. When viruses insert their genetic material into host cells, the host cell itself is forced to express the genes contained in the viruses. The only way to really proactively deal with viruses is with a vaccine. Vaccines are dead or weakened viruses that allow our immune systems to learn to recognize them and start to fight them off at very little risk. Vaccines have to be given before exposure to the virus. They do almost no good if they're given after the virus is contracted, which is why it's essential that vaccines are done proactively as recommended by doctors. A good analogy for how viruses work is found in the section 3rd matrix, if you've seen those by any chance. Agent Smith functions as a virus in these movies. Agent Smith cannot create copies of himself without having a host within the matrix that he can insert his code into. And I think the best place to end any chapter is with a discussion of Agent Smith in the matrix.